translocation, duplication, and inversion. 4 types of chromosome mutation. Sex-linked gene. Sex-linked gene meaning that they are located on the X chromosome. What are the genes located on the X chromosome? For example, the color blindness and also hemophilia, and also a type of muscular dystrophy. They are all on the X chromosome. How many of you cannot read the number on this slide? If you cannot read the number on this slide, then you have that means you have color blindness. Let's take a look at the possible crossing of the different cases of the color blindness. For example, now we have the normal female crossing with the colorblind male. So in this case, you can see that, okay, we have 3 cases. Unaffected individual, meaning that the individual carry only the wild type normal gene, not the wild type normal, the dominant allele. The unaffected individual carry only the dominant allele. Carrier, meaning that the person is heterozygous dominant, meaning that it has 1 dominant allele and 1 recessive allele. And the colorblind individual, that person only has the recessive allele. So let's take a look. How you may or may not have noticed that most of the color blindness that we observe every day, they are mainly male. Why? Because male only need to have 1 recessive allele on the X chromosome, then he will develop a color blindness. For female, they require 2 recessive allele on the X chromosome, then the female will develop color blindness. That's why color blindness has a higher prevalence on male. So when unaffected female, so this is the normal female with the dominant allele. It is a homozygous dominant. Okay, the female is homozygous dominant. So the eggs, they are both the allele in the allele in the gametes. They are both they are both dominant allele. But for the colorblind male, 1 allele is recessive, the other one is just Y chromosome. Okay. So after we cross it, then may I ask what will be the phenotypic ratio of all the children? Okay, phenotypic ratio of all the children. Okay, let me erase this. Sorry. So the phenotypic ratio of all the children, as we can see that it has 2 carrier and 2 unaffected. So the phenotypic ratio will be 50-50% or 1:1. What about the genotypic ratio? The genotypic ratio will be, as you can see that the carrier, they are heterozygous, so it will be it will also be 1:1. Okay, this is all children. What about if we separate the gender? Okay, we separate the gender. What will be the phenotypic ratio for boy? Phenotypic ratio for boy will be 100% normal. Phenotypic ratio will be 100% unaffected. So it will be, let's just say 100% or just 1. What about genotypic ratio for boy? Genotypic ratio will also be 100%. What about the phenotypic ratio for girl? Again, 100%. What about the genotypic ratio for girl? It is also 100%. It seems easy. Let's take a look at the second example. Okay, what is the overall phenotypic ratio of all children? Phenotypic ratio, you will have 2 unaffected, 1 carrier, and 1 colorblind. So it's 2, 1, 1. What about the genotypic ratio for all children? Well, genotypic ratio, we cannot count the genotypic ratio. The reason is that what genotypic ratio? Because the male and female, they are different. It is very difficult to count genotypic ratio. Genotypic ratio, basically 1, 1, 1, 1. We have 1. Yeah, we have 1, 1, 1, 1. So as you can see, this it has on the X chromosome, you have the dominant allele here. So dominant, homozygous dominant here. You have a heterozygous dominant here. But for male, it is different. It is the dominant with Y and then recessive with Y. So the genotypic ratio of all the children, they are 1, 1, 1, 1. Okay, now let's do each gender together. Phenotypic ratio for boy. What is the phenotypic ratio of boy? So you have 1 to 1. That means 50%. What about genotypic ratio for boy? It is also 50%. Okay, now let's take a look at the girl. Okay, phenotypic ratio for girl. What is the phenotypic ratio for girl? Okay, you will have also 1 to 1 or 50%. So 50% of the girl will be normal and 50% of the girl will be carrier. What about genotypic ratio for girl? It is also 50%, 1 to 1. Because you have 50%, half of the girl, they will be homozygous dominant, and then 50% of the girl will be heterozygous dominant. It is when the mother is carrier, the father has a normal vision. The third case is when the mother is carrier but married to a colorblind guy. So what will be the overall phenotypic ratio? The overall phenotypic ratio will be 1, 1, 2. Because you have 1 carrier, 1 healthy unaffected, and then 2 are colorblind. the genotypic ratio is still 1. The genotypic ratio is still 1. Okay, now let's take a look at each gender. The phenotypic ratio for boy, it is the same, 50-50%. The genotypic ratio of boy, well, it's also 1 to 1. What about the phenotypic ratio for girl? Same thing, it is 1 to 1. What about the genotypic ratio for girl? Same thing, it is 50-50%. So as you can see that I intentionally do this so that you know that 50-50% is the same as 1 to 1 ratio. Okay. But in this case, you have 50% of the children, okay, total 50% of children that can be colorblind, okay. Okay, this is the homework exercise. I want you to do the Punnett square to cross a colorblind mother with a normal father. I want you to draw the Punnett square and do it. The reason is that I guarantee that I will test you about this during the exam. So if you watch my video and do your homework, then it will be easier during the exam. If you didn't watch my video and didn't do your homework, then you don't during the exam, then you have to do the Punnett square. It will slow you down. So okay, this is the reward for people who patiently watch my video. That's the end of the genetic lecture. It's very long,